Assalamu alaikum. Good afternoon, ladies and gentlemen. This is Hina Aslam, MPhil scholar from Department of Physiology, Government College University, Faisalabad. It gives me immense pleasure to extend to you a warm welcome to all presented here for the first International Brain and Biomedicine Conference, IBBC 2023. On behalf of organizing committee of IBBC, I am going to moderate this session, which includes different interesting talks related from the field of food science and nutrition, plant sciences, and drug discovery. I feel so honored to be a part of this conference. I have 8 abstracts for all presentations. So let's begin our session with our first presenter. For that, I would like to invite Muhammad Zakiu Kolawal, Adisa Raimi from Concert University, Islamabad. And his title of presentation is Honey and Black Seeds Synergistically Promote Regeneration of Oligodendrocytes in Cuprison Intoxicated Whole Grain. Please, Muhammad Zakiu.
Hello everyone, my name is Mohamed Zakiu Kolawale Adisa Raimi. I'm a PhD candidate biosciences from Benin Republic, and I have a great honor today to present to you my work, Honey and Black Seed Synergistically Promote Regeneration of Oligodendrocyte in Cuprison Intoxicated Coil Brain. So here is my outline. In the central nervous system, we have 2 types of cells, glial cells and neurons. Glial cells are mainly astrocytes, oligodendrocyte, and microglia. They support neuron. Type of oligodendrocyte are myelinating oligodendrocyte and satellite oligodendrocyte. Oligodendrocytes regulate the speed of the action potential transmission, assist the neuron in the axonal transport, maintain their integrity, provide them nutriment and iron. They are highly populated in the corpus callosum, in the subventricular zone, and in the cerebellum. They may be damaged by injury, by toxin exposure, by inflammation, by degeneration. The disorders are dysmyelination, that's leukodystrophy, and this demyelination, that's multiple sclerosis. Multiple sclerosis is an autoimmune disabling disease of the central nervous system and affects 2.8 million people worldwide, mostly young adult people. And the symptoms can be diverse in the human body. In the central nervous system, we can observe like fatigue, cognition, impairment, depression, unstable mood. In the visual, in the speech, in the throat, in the musculoskeletal, in the sensation, in the bowel, and in the urinary. Treatment approaches are so far medication that only help to relieve the symptom. We have stem cell therapy that is very also very expensive, and the choice of a stem cell is very challenging. We have hormonal therapy that also have side effects. And we have natural products that does not have side effects. They are low of cost and they can respond after long term of use. So for this disease, we need a natural product that have anti-inflammatory and antioxidant effect. That's why here we use honey and black seed. Multiple sclerosis model are so far experimental autoimmune encephalomyelitis, toxin-induced demyelinating model, and people use mostly cuprison. And here we try, and all of these models are being used in the mice. So here we try to develop a new animal model for our study. Our new animal model here is Japanese coil. And this animal model has been chosen because it have a large number of offspring in the few time, and the next generation can come after 16 to 17 days. So we aim to estimate the oligodendrocyte population in the demyelinated and remyelinated animal brain to compare the effect of honey and black seed and the mixture of the honey and black seed in the cupridone-treated coil brain to find out the most effective treatment to confirm the amelioration in the behavior of animals remyelinated by honey and black seed. So here is our methodology. We use coil male at 14 weeks old. We have 35 animals that we split in in 7 groups of 5 animals per group, and we mix 200 milligrams of cuprizone with wheat flour to make small ballon that we administered to all of our group for 6 months. Then after 6 months, we then sacrifice the animal of positive control group and negative control group of 6 months to appreciate, to assess the effect of the cuprizone in the damage of oligodendrocyte. And after that, we kept one group without any treatment of honey and black seed for 6 weeks to assess the spontaneous remyelination, and then we administered honey at 100 milligram, black seed oil at 100 milligram, and the mixture at 200 milligram for respectively 3 of the remaining group. So here is our result. Here is the full brain of the coil, and here is the half brain of the coil. Here we have cerebrum, here we have cerebellum, the midbrain. So as our animal model doesn't have a clear defined corpus callosum, we focus our work in the cerebellum. So after 6 months, we did histology of normal brain and the cuprizone-treated brain, and we observed that the thickness of the white matter in the cerebellum fold is decreased in the cuprizone-treated brain compared to normal brain. So we also observe that in the white matter of the cerebrum, oligodendrocyte with branches, and we observe dead oligodendrocyte in the cuprizone-treated brain. Same was also observed in the cerebellum white matter of normal brain, and also in the cerebellum of white matter of cerebellum of the cuprizone-treated brain. Likewise, we also perform body weight that show us at the end of the treatment, we observe 5% body weight loss after 6 months of treatment, and people observe 10% body weight loss using rodent, using also the same dose of cuprizone, and also for 5 weeks. So we thought that the difference may be due to the double number of neurons in the bird than mammal of the same brain size, or maybe due to the dose of the cuprizone we use. We also observed that the body weight has likely been increased in the treated group of honey and black seed and the mixture. So at the end of our experiment, we then sacrificed the animal of the remaining 5 group, and we did histology using hematocillin eosin and count the number of oligodendrocyte population in the white matter using image J. So we noticed that the population of oligodendrocyte has started increasing in the group left for spontaneous remyelination assessment. Also, honey and black seed group and the mixture also show more oligodendrocyte compared to cuprizone-treated group. So here is our result. When we compare the group left for spontaneous remyelination assessment to the group of cuprizone treatment of 6 months, we compare that the population is higher here compared to this group. So meaning that the spontaneous remyelination has occurred. And again, all the group treated by honey and black seed and the mixture also have high population of oligodendrocyte compared to cuprizone-treated group. These results are similar to what people has found assessing remyelination in EAE model in Pakistan animal model using mouse. We also did behavior study, and we observed, like, for that we use beam walk test and fly test. So for beam work test, we score our bar to 0.20 and we left the animal cross the bar and record where they sleep. For flight test, we left the animal flight at constant height and we record the length of the flight. So our results show that all the animals treated by honey and black seed and the mixture have higher score either for beam work test or flight test compared to group treated by cuprizone only. So in conclusion, cuprizone induced oligodendrocyte death after 6 months of treatment. The spontaneous remyelination has occurred after the arrest of the treatment, and honey and black seed improve the oligodendrocyte population regeneration, and even the animal behavior also was restored. But the mixture was more efficient. So in the future, we plan to use a specific antibody to assess all the glial cells involved in multiple sclerosis using same animal model. Thank you for your attention.
Ladies and gentlemen, to know more about advancement in the field of food science and nutrition, I'm going to call our next presenter of the session. That is Arham Javed Malik from Noor International University. And he will share his research work on impact of ketogenic diet on the health status of adults. Please, Arham Javed.
Assalamu alaikum. My name is Arham Javed Malik and I have done my post-graduation from Noor International University, and the topic of my research is impact of ketogenic diet on the health status of adults. These are the highlights that I will be following throughout this presentation. Introduction, introducing ketogenic diet in front of you, objectives of my research, methodology, how this research was planned, results and discussion, what was the result of this research, and comparing them with the previous researches, concluding my research, and then references. Talking about ketogenic diet, keto diet is a high-fat, medium protein, and low-carb diet. It consists of 80% fat, 15% protein, and 5% carbs. Initially, this diet was actually introduced to help in children in the 1920s with epileptic seizures. Additionally, this diet actually shows in the treatment of various diseases that are hormonal imbalances, PCOS, diabetes, UTI. In the year 2020, this diet was the most searched diet globally. It was predicted that by 2027, 15.6 billion people will be following this diet. The leading countries with the most ketogenic followers are USA and Canada. Objectives. The main objectives of the study were as follows. To develop a questionnaire to assess the health impact of ketogenic diet on adults. To determine the reliability of questionnaire on Kronsbeck mark scale. To observe the impact of keto diet on the overall health status of adults. To explore the correlation of ketogenic diet and health outcomes. To assess the differences in health status of individuals following ketogenic diet for various duration. Hypothesis. The hypothesis of this study were: hypothesis number 1, ketogenic diet will likely to have significantly negative relationship with health. Hypothesis number 2, adults following 4 months of keto will likely to have greater health. Methodology. Study location, various institutes like gym, hospitals, and universities. Study design was quantitative method. Study duration was 3 months. Selection of keto followers included males and females of 20 to 40 years of age who were following keto diet for 2 months or more, excluding the people who were following no diet or other diet, or people having any disease or any other alignment or taking medication. Questionnaires were given to be filled and final data was collected. Results. This the sample size for this research was 80, and the age was 20 to 40 years, which had a mean age of 30.50. Mean previous weight of the participants was 88.46, and mean current weight of the participants was 71.57. And this shows that the ketogenic diet had an impact on the weight. Psychometric properties of health and ketogenic diet scale. A reliability of a scale portrays how effective a scale is. This is known as Cron's-Beck alpha, the measure of internal consistency, that is how closely are the items related in a group. I had developed my questionnaire myself, so I had to determine its reliability. I had divided my scale into two groups, health and ketogenic diet. Health had 14 items, which had a Cron's-Beck alpha of 0.70, which is considered reliable. Ketogenic diet had 10 items and had a Cron's-Beck alpha of 0.50, which is also considered reliable. Intercorrelation for ketogenic diet and health status in adults. When we are checking the relationship between two variables, we performed the Pearson product movement correlation. It showed that significant negative relationship between ketogenic diet and health, which is actually proving my first hypothesis that there will be a significantly negative relationship between ketogenic diet and health. Mean differences in months on ketogenic diet and health. One-way ANOVA is actually applied when we are checking the differences between variables of groups. Adults following 6 months of ketogenic diet have greater differences than 2, 4, 5, and 5 months of following keto. So this means that 6 months have a greater difference than 2, 4, 5 months. Which is, and so if you remember my second hypothesis, that was that 4 months will have a greater impact on health. But this result is actually rejecting my second hypothesis. These researchers actually stated that weight loss can be distinguished, but it can raise various side effects for long-term periods. As the body's mechanism is shifted to fats, the body's source of energy is eliminated. As carbs are the sources of your energy, and you are not taking that much carbs to give yourself energy, and you are just gaining energy from fat. The level of urination rises, and the body becomes sustainable to suffer from dehydration. Meanwhile, the body shifts to ketosis, showing the symptoms of keto flu. Keto flu is actually like a normal flu, but you can feel nausea, dizziness, a simple flu, fever with this keto flu. As far as the negative impact of keto diet is concerned, it has been shown that the keto diet causes kidney stones and micronutrient deficiencies, thus affecting human health. Micronutrient deficiencies can be caused as you are not taking fruit, you are not taking the basic nutrients that would give you these vitamins and minerals. So basically your body is being deficient of these nutrients. This researcher reviewed that difference in the duration of the keto diet following. In their study, they targeted athletes following this diet from 21 days. Their data showed difference in the endurance strength as the duration increased, whereas the data in this research was from 2 months till 6 months and revealed 6 months with greater differences. the longer the duration, the advanced the difference will be on the health. Conclusion. My study concluded that that the correlation design actually proved that there was a negative relationship between health and adults. Sorry. One way ANOVA revealed that 6 months had the greater difference than 2 months, 4 months, and 5 months. Multiple people followed this diet on their own. There is a limited number of carbs, so people are tending to eat less than they are craving for. It is advised that this diet be followed under the supervision of a nutritionist. The people should get medical checkups before following this diet. These are my references.
We are moving toward the third presenter of the session. That is Shagufta Praveen from the Government College University, Faisalabad, and her title of presentation is Ascorbic Acid, Benzyl Aminopurine, and Moringa Leaf Extract Induced Drought Stress Tolerance in Pea. Scientific name Pisum sativum. Please, Shagufta Praveen.
Bismillahirrahmanirrahim. Assalamu alaikum. Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Shagufta Praveen from Department of Botany, Government College University, Faisalabad. The title of my presentation is Benzyl Aminopurine, Moringa Leaf Extract, and Ascorbic Acid Induced Drought Stress Tolerance in Pea. Benzyl Aminopurine, BAP, it is an important plant growth regulator that affects cell division, chlorophyll biosynthesis, photosynthetic efficiency, and nutrients uptake. BAP has very important role in the plant growth and development. For example, different studies show that when BAP is applied exogenously as foliar spray or as priming agent, it can increase photosynthetic rate, yield, and different metabolites under different levels of stress. For example, in cotton, under 50% reduced irrigations, foliar application of 5 micromolar BAP can increase photosynthetic rate under 175 and 50% field capacity. BAP at the rate of 50 milligrams per liter can increase crop yield in maize under 140% field capacity. BAP at the rate of 50 ml per pot can increase ascorbic acid and total phenolic contents in maize. Moringa oleifera. Moringa oleifera is a fast-growing drought-resistant tree that belongs to family Moringaceae. The leaf extract of Moringa oleifera is rich in mineral nutrients, growth hormones, vitamins, and antioxidants. Various phytohormones include auxin, gibberellins, Z-trim type cytokinins. MLE increase seedling establishment, plant growth, eventually production in the field crops. It increases leaf area, plant height, biomass, and chlorophyll contents in crops. Ascorbic acid, that is also known as vitamin C, can detoxify reactive oxygen species and improve growth of plants under different abiotic stresses. It functions as major redox buffer, regulate different metabolic processes like photosynthesis, hormone biosynthesis, and regeneration of other antioxidants. Foliar application or as priming agent in both ways. Exogenous application of ascorbic acid is effective in increasing the germination rate, shoot and root length, shoot and root dry rate under different levels of stress. For example, under 114-50% field capacity, Ascorbic acid, when used as priming agent, can increase growth of Brassica napus and increase shielding peak and improve growth in sun. Heat, that is commonly known as murder, is an important member of family Leguminosae. It is the second most important food crop that is extensively grown in temperate regions. It is the growth season of the tea is October to November. Tea is rich in proteins, carbohydrates, vitamin C, magnesium, and phosphorus. When used in 100 grams edible quantity. Drought stress. Water is one of the essential factors for the sustainability of life of all living organisms, including plants. Plant development and yield is influenced by direct or indirect effects of climate change. It has been reported that annually 55 million people worldwide are affected by water deficit stress. Water deficit stress can lead to 50 to 70% crop yield reduction globally, according to a survey of World Health Organization. It is estimated that by 2030, about 700 million people, that are about 40% of the global population, will be at the risk due to water stress. Effects of drought stress on plants. Drought stress brings about different morphological, physiological, and biochemical changes. These morphological changes include increase in the number of leaves, leaf area, increase in root length, leaf aging, changes in growth season stage. Various physiological changes include transpiration, stomatal activity, photosynthesis, osmotic balance, water transmission, and relative water content. Biochemical changes includes modulation in the antioxidant defense system, photosynthetic pigments, accumulation of compatible solutes that include proline, glycine, hormonal contents, and secondary metabolites. Plants cope drought stress by adopting different mechanisms. These mechanisms may be stress escape, avoidance strategy, and tolerance mechanism. Stress escape mechanism is due to the shortening of the life cycle and by plant development. Avoidance strategy is the retention of optimum relative water content and high water usage. Plants' water potential is maintained high through a reduction in the stomatal transpiration and the increase of water uptake from well-established sources. Tolerance mechanism, that is a survival mechanism, it includes xeromorphic characteristics such as the presence of hairy leaves and cuticles that may help to maintain high water potential in plant tissues, osmotic adjustment, and solute accumulation. Hypothesis of the current study was to check whether or not the foliar application of DAP in combination of moringa leaf extract MLE and ascorbic acid SASA help in mitigation of drought stress of plants. Objectives were to identify the effect of DAP, MLE, and ascorbic acid on the growth, yield, and physiology of plants under drought stress. To investigate the pattern of changes in terms of growth and physiological and biochemical parameters of tea plants when foliarly applied with mixture of DAP, MLE, and under drought stress or stress conditions. Materials and methods include pot experiment was conducted at the Botanical Garden, Government College, University of Hyderabad. There were two pea cultivars, that is Meteor and P09. There were two drought stress levels, that is 100% field capacity and 60% field capacity. There were 5 foliar treatment levels, that is control, water spray without any treatment, BAP 0.005%, MLE means moringa leaf extract, that is 30%. Ascorbic acid, that is 0.005%, and then a combination of DAP, MLE, and ascorbic acid in 1:1:1 ratio. Findings. The results of the current study shows that shoot and root fresh weight significantly decreased under 60% field capacity in both pea cultivars, that is Meteor and P09. Of both pea cultivars, P09 was higher in shoot fresh weight and root fresh weight. Foliar application of DAP, MLE, and ascorbic acid significantly increased shoot fresh weight and root fresh weight in both pea cultivars of various treatments. Mixture means combination of DAP, MLE, and ascorbic acid was more effective in increasing the shoot fresh weight and the fruit fresh weight in both pea cultivars. Chlorophyll contents, chlorophyll A and B contents significantly decreased under 60% field capacity in both pea cultivars of various treatments, PAP, MLE, and ascorbic acid. A mixture of PAP, MLE, and ascorbic acid was more effective in increasing the chlorophyll P contents in the pea cultivar P09. Valendialdehyde and hydrogen peroxide contents significantly increased under 60% field capacity of both pea cultivars of various treatments, mixture of both of various treatments, the BAP, that is 0.005% BAP, MLE 30%, and ascorbic acid 0.005%. All these three treatments significantly reduced the NDA and contents in both pea cultivars, particularly in Meteor. Activities of antioxidant enzymes like superoxide dismutase and peroxidase significantly increased under 60% field capacity in both pea cultivars. Various treatments, BAP, MLE, and ascorbic acid significantly enhanced the activities of SOD and POD in both pea cultivars under stress or low stress conditions. Catalase activity significantly increased under 60% field capacity and by foliar application of PAP, MLE, and ascorbic acid. Proline contents significantly increased in both pea cultivars under 60% field capacity. All treatments significantly enhanced proline contents in both pea cultivars under non-stress and low stress conditions. However, MLE 30% was more effective in increasing the proline contents in the pea cultivar Meteor. In conclusion, the foliar applied BAP, MLE, and ascorbic acid are proved as growth promoters of pea plants under drought stress. These growth promoters alone or in combination can be effective in increasing the enzymatic and non-enzymatic activities of antioxidants under drought stress of cultivars. While combined application of BAP, ascorbic acid, and MLE was considered as more effective strategy under drought stress on pea plants. Thank you.
Thank you for such an appreciable talk. It's time to encourage the next presenter, which I'm going to call is Asim Masood from Peer Mihir Ali Shah Arid Agriculture University, Rawalpindi. He will share his research views on preservation and importance of vitamin C in fruits and vegetables. Please, Asim Masood.
Hello everyone. Hello everyone, this is Asim Masood and I'm from Institute of Food and Nutrition Science, Peer Mihir Ali Shah Arid Agriculture University, Rawalpindi. Today I'm here in IBBC International Brain and Biomedicines Conference 2023 to present my topic, that is preservation and importance of vitamin C in fruits and vegetables. So here I'm going to start. First of all, I will give a short introduction about vitamin C because here, according to my topic, I'm talking about vitamin preservation in fruits and vegetable. So here I'm going to give an introduction, a brief introduction of vitamin C and its importance. Vitamin C or ascorbic acid is a water-soluble vitamin and it is very essential for our body. It is essential with health respect, but if we want to store our product or if we want to store our commodity for a long period of time, vitamin C or in other words, we can say like ascorbic acid is very important. Even before its discovery in 1932, nutrition experts recognized that something in citrus fruits could prevent scurvy, a disease that killed as many as 2 million sailors between 1500 and 1800. This is the historical importance of vitamin C. Means a citrus in citrus it is also present, and it can cure disease, or it can be preventive against the disease which is known as scurvy. So here it is a historical reference also present. Next, I'm going to tell that foods that contain a high amount of or high content of vitamin C. Among those, there is rose hips, sea buckthorn, red pepper, papaya, kiwi, cauliflower, guava, celery, spinach, melon, cabbage, pineapple, etc. There are many commodities in fruits also and vegetables that contain vitamin C. Further, health benefits of vitamin C. As in the introduction, I've also given you the historical reference in which in 1500 to 1800, many of the sailors were died because of scurvy disease. So here, some of the health benefits of vitamin C is that it helps managing blood pressure, helps preventing in iron deficiency, may reduce the risk of stroke, help with better skin aging, reduce the risk of cancer and cardiovascular disease, reduces inflammation, protects your memory. Further, that reduction of vitamin C during storage. We know that when a commodity is harvested, for example, if I say that a fruit is harvested and it is brought to the storage chamber, then what happened during with the course of time, the vitamin C content start decreasing. Here you can see in the graph that the vitamin C content is going on decreasing. With the decrease in vitamin C content, or you can say with the decrease in ascorbic acid content, the fruit commodity or the vegetable commodity faces many of other negative impacts also, like they shrink and the water is lost, their economic value decreases. That I'm going to talk further in the next slides. During storage, the vitamin C content gradually decreases at a rate depending on the processing and storage temperature. The more rapid decrease of ascorbic acid concentration at the beginning of the storage can be attributed to the immediate reaction of an amount of ascorbic acid with the dissolved oxygen. Here, in order to store, if you want to store a commodity, for example, I say that I'm going to store citrus, or I'm going to store mangoes, or I'm going to store some sort of vegetables. So I have to maintain a temperature, humidity in which vitamin C remains in the content. It lost, but it lost in a slow rate, not in the fast rate. Further, why vitamin C is lost in fruits and vegetables. So in the previous slide, I talked when the temperature is not managed properly, when the storage atmosphere is not managed properly, when like we do blanching in our food science. Blanching is the treatment in which we subject the fruits or vegetables in the hot water to kill the microorganisms. So vitamin C also lost in the blanching. Then bruising, abrasions, means a cut or trim can also cause a loss in the vitamin C content. Then electromagnetic energy also cause loss in vitamin C content. So these are the parameters, or these are the characters that you can say aids in the loss of vitamin C more faster. Further, why vitamin C is important in storage stability of commodity. Here, according to my presentation, my whole you can say inclination was on this thing, that why vitamin C is important in storage stability of commodity. We face food security problems in our country, so that's why it is necessary to keep our commodity or keep our product keep the shelf life of our product enhanced, in order to you can say in order to stable our food security or food safety. So that's why vitamin C is important. If the vitamin C, because it is the principal, you can say that it is the principal storage index. If vitamin C is conserved in the commodity, all the other components also remain conserved in the commodity. So that's why you can say that vitamin C is very important. Like vitamin C is conserved, so you can say other volatiles or other commodity components like minerals, etc., other phenolic compounds, means all the necessary components remains viable if the vitamin C is viable. Further, what happened when vitamin C is lost in fruit and vegetable? So number one, commodity gets shrink. Here you can see in the image also that I have posted an image of apple in which a commodity gets shrink. It gets dehydrated, it gets loss of the water, and ultimately to the loss of the commodity. Volatiles are formed means smell start increasing in the product. Degradation of different polymers start, different polymers like starch, you can say phenolic compounds, you can say cellulose, you can say lignin, all starts degrading. Then economic value decreases. Economic value of the commodity also decreases. Why decreases? Because if I talk about an individual person, you are not going to buy a commodity that is not aesthetically attractive to your eyes. That's why you're not going to buy that commodity. So you can say that when vitamin C is lost, so commodity faces a huge problems. Further, how to preserve vitamin C in fruits and vegetables. Now, this is also an important topic, how to preserve vitamin C in fruits and vegetables. First of all, control atmospheric conditions. We have to control the atmosphere. Now, what are control atmospheric conditions in our food science and technology? Control atmospheric conditions are basically those technologies which are applied in a storage chamber. Here, the concentration of oxygen decreases and we increase the concentration of carbon dioxide. Like you can say that the concentration of oxygen decreases to 10% and carbon dioxide increases to 2 to 3%. So that's why there's a minimum loss of vitamin C in fruits and vegetables. Then temperature management, if we manage the temperature. Now, temperature vary from commodity to commodity. Okay, different commodities have different favorable temperature, so that's why temperature management is very important. Humidity control is very important. Mostly products you can say that are perishable, that contains water in, like that are, you can say, succulent commodities. They have a large amount of water, or you can say a high amount of water. So that's why humidity control is very important. Otherwise, there could be possible fungus attack. There could be possible loss of vitamin C or any other component. Then harvesting at early hours. Now, what is this? This is that when a commodity is harvested in the field. So usually the fruits are mainly in Pakistan. Most fruits are harvested in summer season, like mangoes. Mangoes are our major cash crop, so it harvested in summer season. So the field heat can also damage the crop. So that's why it is recommended that we should harvest the crop in early hours, like in the morning or in the evening. Then compartmentalization in storage. Now, this is also a very important point, compartmentalization. Now, what is compartmentalization? It is that in storage chamber, there must be different compartments for different commodities. In one should not place every commodities in same compartment, like guava, apple, peaches. They should be kept in different compartments because they are different commodities. Their characteristics are different and they require different kind of treatment. So that's why they should be kept in different compartments, not in the same one. So that's all from my side, and I'm very happy to be the part of this conference. So thank you, everyone. And if you have any question, any query, you can ask. Okay. So I'm very happy and bless you all.
Thank you for sharing your research work with us. Ladies and gentlemen, next abstract is going to present by Bushra Inam from Kohat University of Science and Technology. And the title of her presentation is Isolation of Hydroxyapatite from Fish Bone for PEG-based nanocomposite films prepared by photocross-linking. Please welcome Bushra Inam.
My name is Bushra, doing MPhil from Kohat University of Science and Technology. First, today I am here to present my topic in International Brain and Biomedicine Conference. The presentation topic is Isolation of Hydroxyapatite from Fish Bone for PEG-based nanocomposite film prepared by photocross-linking. Hydroxyapatite, the chemical formula has been widely studied since 1926. Hydroxyapatite is naturally occurring mineral of biological importance. Human and animal bones are composed of hydroxyapatite. Hydroxyapatite is considered as biomaterial widely employed in many health applications. Because of its chemical property, hydroxyapatite exhibits a great bioactivity and is highly compatible with digestion, bones, and teeth in living beings. Heart tissue restoration uses HA in many ways, although not in its purest form. Pure HA has pure mechanical quality that make it unsuitable for load-bearing application. To broaden its application, HA is typically added to composite or polymers. Polyethylene glycol-based material are widely studied for biomedical application due to their beneficial qualities such as durability, minimal immunogenicity, and easy to use. Hydroxyapatite has been utilized in dentistry to replace missing teeth. Along with dental cement and filling, toothpaste also include HA. The use of HA in medicine delivery is another instance. HA is a strong candidate for use as a drug carrier because of its inherently porous structure and high binding affinity, which creates a niche for drug loading. Nanocomposite, the application of HAP nanoparticle or nanocomposite are wide as discussed earlier. However, it was observed that the stability, mechanical and physical properties of HAP-based materials can be enhanced by combining it with PAG. The objective of this presentation is isolation of hydroxyapatite from fish bone for PAG-based nanocomposite film prepared by photocross-linking. Experimental work: first of all, we separate hydroxyapatite from fish bone and then we use in clinical application. Isolation of hydroxyapatite: first of all, we separate bone from fish and then after treated with chemicals, and after we that separate through calcination process, and we separate hydroxyapatite. Isolation of hydroxyapatite: for the alkaline hydrolysis procedure, the bone of roe will be boiled for 15 minutes at room temperature to completely remove the meat, and the fish bone will be treated with sodium hydroxide at 250 degrees centigrade for 5 hours to ensure the organic moieties are properly removed. Once the pH neutralized, the mixture will be filtered through a filter paper using continuous water washing. At 250 degrees centigrade, the product will be dried in an oven. Fish bone will be put in silica crucible and heated to 1900 degrees centigrade in a muffled furnace as a part of the thermal calcination. PG-based film prepared by photocross-linking. To prepare blank film, algacure will be added to PEG-DMA. At the same time, another film will be added algacure in PEG-DMA and add hydroxyapatite nanoparticle in solution and kept under the UV light for 3 hours. The cross-link gel will be formed after UV light. The gel will be kept for 2 hours in ambient temperature to completely solidify. After the gel will be washed with distilled water. Characterization: for confirmation, the synthesized hydroxyapatite and prepared film will be evaluated by FTIR and XRD. FTIR is a change in chemical composition of hydrogel with and without HA were determined with FTIR. FTIR analysis of PEG-DMA was performed to characterize and confirm the chemical cross-linking among the different PEG-DMA hydrogel after free radical polymerization. PEG-DMA mixture before polymerization, the largest number of carbon-to-carbon double bond resulted in the highest absorbance peak in the spectrum. After UV polymerization, carbon-to-carbon bond, which disappeared during the curing process, making the success of the free radical polymerization reaction carried out, and the absorbance peaks disappeared because of the conversion of the carbon-to-carbon double bond into carbon-to-carbon single bond. XRD result: the hydrogel without HA and had only one broad peak. With the addition of HA, the height of this peak significantly decreased. It indicated the presence of HA crystal could interfere with the packaging of the PEG-DMA based polymer chains and result in decreased crystallinity in composite scaffolds. Thank you very much.
Thank you so much, Bushra, for such an outstanding presentation. I'm going to call the sixth presenter of the session, that is Maria Maqbool from Government College University, Faisalabad. She will tell us more about protective potential of mentha piperita extracts on retinol-induced motor deficits in mice. Please welcome Maria Maqbool.
Assalamu alaikum, all of you. I am Maria Maqbool from Government College University, Lahore. The title of my research is Protective Potential of Mentha piperita Extracts on Rotenone-Induced Motor Deficits in Mice. First of all, I will briefly explain about motor deficits. Motor deficits or dysfunctions are particularly defined as loss or damage of uncontrolled and coordinated movement in a person's body. These motor dysfunctions or deficits are strongly related with the different neurodegenerative diseases. As they are serious issues, these issues are related with Parkinson's disease, as Parkinson's disease involves the loss of coordinated and controlled movement in the body. Motor deficits are caused by several environmental as well as genetic factors, but the experimental animal models can also be generated with the help of the rotenone. Rotenone is a neurotoxin, plant-based neurotoxin that induces motor deficits in an animal model. This was firstly used for research purpose in 1980s. This is the pictorial diagram uh, of a person who was suffering from motor deficits. In this diagram, we can see that there are tremors in the hands and legs of the person, whereas the elbows and the knees of the person are flexed, and uh, the person's back is rigid, and he cannot maintain its straight body posture, as well as he is unable to walk in a proper way. Next, Mentha piperetta. I have used a Mentha piperetta for my research purpose in order to check its protective role against the motor deficits. Mentha piperetta is commonly known as peppermint, whereas it is commonly known as pudina in Pakistan. It is an aromatic perennial herb with deep green colored leaves and light purplish or pinkish flowers on it. It is used in several synthetic products due to the presence of menthol in it. Its decoction is widely used as a drink. It has several medicinal properties, such as it has antioxidant activity, antimicrobial activity, anti-cancer, neuroprotective, anti-inflammatory, hepatoprotective, and also have anti-allergic activity. Aims and objectives of my study was to investigate the protective effect of Mentha piperetta through the preparation of Mentha piperetta leaves and root extract, whereas the development of rotenone induced motor deficits in mice model, as well as the assessment of motor functional deficits in Mentha piperetta treated as well as in untreated mice. The methodology of my research work was firstly include the preparation of Mentha piperetta leaves and root extract. For this purpose, I dried the leaf and root of the Mentha piperetta plant separately, and then ground them into a fine powder. After this, the powder was mixed with methanol in the ratio of 1:10, and the flask was placed on flask orbital shaker for the period of about 24 hours. After that, the solution is filtered, and the filtrate is placed on the water bath at 42 degrees Celsius for about 12 hours. After that, I lyophilize the Mentha piperetta roots and leaf extracts. Next one is the experimental design and grouping of animals. My experiment consisted of total 28 days, in which I firstly treat the animals with the animals include the pre-treatment, in which I treat the animals with Mentha piperetta extract without the administration of rotenone, while the next 21 days included the post-treatment with Mentha piperetta extract along with the administration of rotenone. I took a total 30 male albino mice and divided them into 5 groups. The first one is the control group, which was only given 2.5% DMSO dissolved in sunflower oil at the dose of 10 ml per kg. It was injected subcutaneously. Next one is the rotenone group. Rotenone group was given 2.5%, 2.5 mg per kg dose of rotenone. Rotenone was dissolved in 2.5% DMSO and 1 ml sunflower oil. Rotenone was also injected as subcutaneously. The other treatment, 3 treatment groups, that is the Cinemet plus rotenone group and the methanolic Mentha piperetta leaves and the root groups, were also given rotenone subcutaneously after 1 hour of the oral dosing. The Cinemet group was given Cinemet at the dose of 20 mg per kg orally, while as the Mentha piperetta leaves and root extract were given the extracts at 200 mg per kg orally. And all these groups were also given rotenone subcutaneously. The next are the behavioral assessment. After this, I assess the animals behaviorally. For this purpose, I performed different behavioral tests. The first one include the narrow beam walk test. This is the apparatus of narrow beam walk, which was consisted of a wooden rod, and the length of the rod is 1.5 meter, and it is 10 millimeter in diameter. This wooden rod is placed at 60 centimeter height, and it was placed, both the stands was placed at the two ends of the rod. The animals were taken from their cage and placed at one end of the rod and allowed to cover the distance of 1 meter under the stress. After this, the time was noticed. All the activities of the animals were recorded with the help of the video camera, and 2 trials were given to those animals. 2 trials were given to every mice. After this, their average reading was taken. This is the graph which shows the time taken by different groups. This shows that the control group took the average time of about 9 seconds, which was the lesser time as compared to the other groups. While if we look at the treatment groups, that is cinnamon, rotinone, as well as cinnamon leaf extract and the root extract group, these shows the time which is nearly equal to the control group. While if we look at the rotinone group, this group took the average time of about 35 seconds, which is the time greater than the other treatment as well as the control group. This type of behavior shows the severe motor deficits, while the treatment group shows the recovered symptoms. The next one is the pole climb down test. Pole climb down, this is the apparatus of the pole climb down test. It was performed in order to show the stiffness and slowness in the movement of animals. Animals were placed at the top of their pole, and then they are allowed to descend from the pole. Their activities were recorded with the help of the video camera, and the time taken to descend from the pole was noticed. This is the graph which shows the time to descend from the pole. The control group was given 9 seconds, while the treatment groups were shown the 11, 12, and 13 seconds. This is the average time. All these time were very close to each other as well as the control group. While if we look at the rotinone group, the rotinone group took the average time of about 25 seconds, which shows that this group shows the severe form of motor deficits, while the other treatment group shows the recovered motor symptoms. The next is the tail suspension test. Tail suspension test was performed in order to check the immobility time of the animals. For this purpose, the animals were hanged at the wooden apparatus, which consisted of 4 chambers of equal size. The animals were hanged for about 6 minutes, and their activities were recorded with the help of the video camera. After this, the immobility time of the animals were recorded, and the graph was plotted. This is the graph which shows the immobility time of different groups. Control group shows the 90 seconds, whereas the cinnamon as well as the leaf extract group shows the lesser immobility time. Whereas root extract show a little deviation, and it shows the average time of about 150 seconds. But this is lesser than the rotinone group. Rotinone group, the average showed the average time of about 230 or 240 seconds. This type of behavior shows the severe motor deficits. Next test is open field test. Open field test was performed in order to check the exploratory rate of the animals when they allowed to move in an open field under no stress. This apparatus consists of a wooden chamber of which is of equal sides, and all the activities of the animals were recorded with the help of the video camera. This is the time which shows. This is the graph which shows the activity of the animals and number of entries in an open field. Because the open field apparatus consists of 16 small squares, this is the number of entry in each small square. This shows that the rotinone group shows the lesser number of entries as well as the other treatment groups. This shows that the rotinone group shows the motor deficits. These are the trajectories. The trajectories of the mice were also drawn in an open field. This is the trajectory of the control group, which shows that the mice covered the greater distance when it allowed to move in an open field, while the rotinone group showed the lesser distance covered, as well as it shows that the mouse only moved along the boundaries of the apparatus. This type of behavior showed motor deficits, while the cinnamon, leaf extract, and root extract group showed the greater distance covered by the animals. This type of behavior showed the recovered motor symptoms. Next is the conclusion. Conclusion of my research was that motor deficits were observed in rotinone group, and oral dosing of mentha piperita extracts recovered the motor impairments in treatment groups, whereas mentha piperita can also be considered as potential therapeutics for further investigations. Thanks to all of you.
Thank you for deeply impressing the importance of this project. Now I would like to call the next presenter, Muhammad Hasnan Latif from University of Veterinary and Animal Sciences, Bahawalpur. And his title of presentation is Determination of Meat Quality, Parameters and Sensory Evaluation of Grey Franklin in Punjab, Pakistan. Please, Muhammad Hasnan.
Assalamu alaikum. The author of this presentation is Muhammad Hasnain Latif from Cholistan University of Veterinary and Animal Sciences, Bahawalpur. For some genuine issue, he cannot here to present the oral presentation. My name is Virisha Saif from Cholistan University of Veterinary and Animal Sciences, Bahawalpur. I am the member of this presentation, so I will represent. The topic of presentation is determination of meat quality parameters and sensory evaluation of grey francolin in Punjab, Pakistan. Introduction. The grey francolin is a game bird that belongs to family Phasanidae of order Galliform. It is a palearctic species that may be found mostly in temperate zone. Grey francolin is being produced professionally for meat production all over the world. Their meat comprises 25.2 to 29.1% protein, 11.2 to 1.4% ash, 55.9 to 62.4% moisture, 1.6 to 5.6% fat, and 72 to 34 milligram cholesterol, indicating that it has the ability to produce excellent meat. Their meat, particularly the breast muscle, has a high protein concentration and low fat content. Material and method. Study area. The present study was conducted to evaluate the meat quality parameter of grey francolin collected from 6 different localities of Punjab, Pakistan. Total 12 samples, 6 male and 6 female of grey francolin was collected from different sites of Punjab, which are Bahawalnagar, Bahawalpur, Hasipur, Khanewal, Melsi and Vihari, from the registered breeder under Punjab Wildlife and Parks Department, Government of Punjab, Pakistan. Meat quality parameters. There are 4 meat quality parameters. First one is determination of moisture content and dry matter. Second one, determination of crude protein. Third one is determination of crude fat, and fourth, determination of ash content. From their formula, we can identify or determine meat quality of grey francolin. Sensory evaluation. Sensory evaluation was performed by comparing the meat of grey francolin with the meat of chicken. For this purpose, semi-trained panelists were recruited for using scoring performer methodology. A panel of 10 members was recruited from faculty members of Department of Zoology, Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Sensory descriptor was defined for the parameter of aroma intensity, aroma desirability, juiciness, tenderness, palatability intensity, and palatability desirability, and overall acceptability. The evaluations were made on the same day after preparation of sample. After meat thawing, chest fillets and thigh were cooked. Meat of grey francolin and chicken was cut into bite-sized cubes and was not seasoned or salted. All members were expressed to one significant digit on 1 to 10 points numerical scales, with a higher score indicating higher attribute intensities. Consumer panelists were served with one sample at a time and instructed to clean their plates between samples. 10 points hedonic scales were used to assess overall liking and liking of appearance, texture, and flavor. For sample of here is the picture of grey francolin. Statistical analysis. On the completion of samples collection and laboratory analysis, data was tabulated and statistical analyzed. Unpaired t-test was applied to determine meat quality of grey francolin using SPSS 22 statistical software. Results. The overall data of body composition parameter was compiled, in which body weight, carcass weight, dressing percentage, neck weight, wing weight, breast muscle weight, leg muscle weight, and fat content of grey francolin was measured. Highly significant variations were prominent in body weight, while significant variations were noted in neck weight of both sexes of grey francolin, and all other parameters are non-significant. The overall data of sensory properties were compiled in which aroma intensity, aroma desirability, juiciness, tenderness, palatability intensity, palatability, desirability of grapefruit was compared with chicken. The comparison between grapefruit and chicken in aroma intensity, aroma desirability, juiciness, tenderness, palatability intensity, and palatability desirability was highly significant. Therefore, it is calculated that no variation in body composition was recorded except body weight, neck weight in both sexes of grapefruit. The highly significant variations among the sensory qualities attribute between grapefruit and chicken show the customer preference due to better sensory qualities attributes in grapefruit. Comparison between body composition of male and female grapefruit. Conclusion. The highly significant variation among the sensory qualities attribute between grapefruit and chicken show the customer preference due to better sensory qualities attribute in grapefruit. These factors will also help us to develop designer and value-added meat products, making grapefruit meat production even more economical and beneficial for human health. Desired meat composition, product type, and quality can be achieved through the above discussed factor. Thank you.
Thank you for sharing such wonderful information and informative research. Ladies and gentlemen, it's time to call the last presenter of the session, and our last abstract is going to present by Dr. Madiha Arshad from University of Education. And her title of presentation is on protective potential of pomegranate extract in nickel intoxicated mice. Please welcome Dr. Madiha Arshad.
Welcome to International Brain and Biomedicine Conference 2023. I am Dr. Madiha Arshad from University of Education, Faisalabad campus, and today I am going to present protective potential of pomegranate extract in nickel intoxicated mice. The branch of science which deals with the toxicant and poisons and their effects and treatment is called toxicology. And the toxicant is a substance that can produce deleterious conditions on a living system, and they may be man-made or naturally occurring. There are some general toxicants such as drugs, radiations, chemicals, heavy metals, and infectious agents. Nickel is an element that naturally occurs in earth crust and its core. It occurs in drinking water, plants, soil, sediments, airs, and various foods. And anthropogenic procedures also contribute nickels to the environment. Nickel is a silvery white heavy metal which is hard and tactile, easily magnetized, good conductor of heat, and exhibits shine and glow. It is a transition metal which has two oxidation states. It is highly resistant to corrosion. That is why it is used for electroplating to make alloys. It is also benefit for some reasons. That is, it is an essential micronutrient for organism's body, important for plant growth, important for nickel metabolism, nitrogen metabolism in plants, important commercial elements in industries, disease resistance in plant. It, on the other hand, it also causes allergic reactions, burning skin rashes, causes lung fibrosis and anemia, enhanced carcinoma formation in rodents, respiratory illness, and tubular necrosis and eosinophilia. It can be exposed via different routes such as dermal contact, inhalation, absorption through skin, gastrointestinal ingestion. It can cause DNA damage via histone methylation. It produces oxidative stress by protein impairment, lipid peroxidation. It also stops DNA repair mechanism by yielding oxygen-free radicals that causes a protein debasement. It decreased concentration of hepatic glutathione in livers and other cells. It can change configuration of DNA binding proteins, cause change in transcription factors of genes. In this study, we tried to find some natural foods that can ameliorate the effect of nickel toxicity. So we use pomegranate for this purpose. Pomegranate helps in digestion. It is widely cultivated in Asia, Africa, and Western areas. It is a drought-tolerant plant and can be grown in dry areas. Its pulp and peel contains phytochemicals. Seed oil contains palmitic acid, stearic, oleic, and linoleic acid, which are good for the heart functions. Pomegranate can decrease oxidative stress, effective against cancer, diabetes, Alzheimer's diseases, anti-atherosclerotic activity, anti-inflammatory activity. It can reduce fatty liver acid in obese people, 2 to 3 times larger antioxidant ability. The main objective of this study was to evaluate toxicological effects of nickel on liver and heart. Its alteration produced by in LFT and lipid profiles, and mainly to check the protective potential of pomegranate peel extract. Nickel fluoride LD50 was taken from the Siam 2008. According to him, its LD50 is between 16 to 113 mg per kg. So sublethal doses was prepared to administer to the mice. There was different groups, for example, control group, which was not received any treatment. Those groups were further divided into 3 groups, which were given 8 mg per kg of nickel chloride, 15 mg per kg of nickel chloride, and 30 mg per kg nickel chloride. Similarly, third group, which was the antidote group, was given with 500 mg per kg to one subgroup as antidote group. Similarly, antidote plus dose 1, dose 2, and dose 3 was also administered in this group. Animal use was male Swiss albino mice, Mus musculus, which was left in steel cages of size 8 into 14 inches. 12 hours standard light and dark conditions was provided. Humidity was maintained at 40 to 45 degrees percentage. Food, chick feed was given to the mice. Water was given ad libitum. In this study, nickel chloride was used as chemical, which was given orally with distilled water for 1 month, 30 consecutive days. Pomegranate unica granatum was administered after 1 hour of dose administration in 500 mg per kg through oral gavage. For peel extract formation, we used fresh peel 50 gram plus 250 ml distilled water, which was boiled for 25 minutes and filtered by using Whatman filter paper, stored in sterile bottle at 4 degrees centigrade. Total sample size was 80 mice, which was n is equal to 10 in 8 groups. Ethical consideration was taken into account, which was according to the ethical committee, provision of clean water, chick feed, temperature maintenance, humidity maintenance, standard light and dark conditions maintenance, and animal safety measures. Similarly, during dissection, no anesthesia was used, rather it was dissected through cervical disjunction, and laboratory preventive measures were also taken into account. For 30 days, nickel chloride and pomegranate extract was given. On 31st day. No treatment was applied to anyone, and that was the acclimatization period. On 32nd day, dissection was done, and blood was collected for biochemical testing, and tissues of livers and heart was also separated for histological examination. Histological testing was done by slide preparation and hematoxylin staining. For biochemical testing, EDTA vials was used to preserve blood. Data analysis was done with SPSS 21 software. One-way ANOVA applied for mean comparison and significant difference between control and dose group were measured. Results: biochemical analysis, lipid profiles in dose group which were given only nickel chloride showed significant increase in cholesterol, LDL, triglyceride, and very low lipoproteins. While in dose plus pomegranate groups, they tend to be decreased or to be in normal range. Similarly, liver functioning test also showed those group which were received dose only nickel chloride only, they had increased bilirubin, ALT enzyme and ALP enzymes were also increased in those dose groups which was administered nickel chloride only. While those groups which received nickel chloride along with the pomegranate peel extract, they showed almost normal bilirubin, ALT, ALP enzymes. At histopathological level, histopathology of heart, in first picture you can see normal intercalated disc, myocytes, myocyte nucleus, while in the second, third and fourth picture you can see the deterioration caused by the nickel chloride, which is intracalated disc degeneration and tubular dysgenesis, while in the antidote group, the histopathology showed normal heart histology. Histopathology of liver also showed first picture you can see normal hepatocyte, central vein, sinusoidal spaces, while in second, third and fourth picture which was treated with the nickel chloride showed necrosis, fibrosis and adenomyosis, vacuolization was seen. While those groups which received pomegranate peel extract with the nickel chloride, they showed almost normal histopathology of liver. Conclusion: nickel chloride caused a high degree damage to liver and heart tissues of albino mice, while pomegranate reduced toxicity and protect tissues from damages at biochemical and histological level. It is recommended to avoid nickel chloride through water, crops, food and food. Pomegranate pulp extract and juice must be used where nickel concentration in the food is high. Organizations, agencies, institutions must spread awareness through modulate rules and regulations, programs and pamphlets. While there are some certain limitations that cannot be avoided, for example, nickel can be consumed through leaching, biological weathering, which cannot be avoided naturally. Chemical, metallurgical industries, metal plating consumed as catalyst, entering in water, soil, air is unable to stop or control. So it must be limited by controlling preventive measures against heavy metals. Thank you so much for your attention.
Thank you so much for your participation and delivering such a wonderful research work. Ladies and gentlemen, all the presentations for this session has ended. I'm thankful to all the presenters deliberating in various fields of sciences. All these constructive innovations gave us new perspective of significant advancement in the field of sciences. Dear presenters, all of your presentations will be uploaded on official YouTube channel. After their evaluations, awards will be provided to all winners of IBBC 2023. All the presentations was highly interesting and informative. Thanks for your participation and attention. Our next session will start very soon. It will include talk from various fields. It will be moderated by one of our colleagues. So stay tuned and stay connected. Allah Hafiz. Thank you.